So we'll have our last mentor talk for the day by Mohan Balasubramanian, who has come all the way from the UK, from the University of Warwick.
Everyone. So first of all, let me start by thanking the organizers for giving me this opportunity to talk in the YIM. This is the first time I'm coming to the YIM. I'm from the University of Warwick in UK, and I'll talk. I think this is kind of the general message, which is curiosity over metrics, and questions. Great questions drive scientific progress. So I want to start with this. How many of you know who this is? Okay, great. And who that is? The batsman, yeah. So that's actually Basil de Oliveira. This is one of the greatest umpires, Dicky Bird. The reason I'm showing this is when I was in school, I used to line my whole wall, like lots and lots of cricket pictures. And so I had only two aims. One is to get into IIT, and the other was to play as much cricket as possible. So I didn't get into IIT, didn't get the greatest scores in my 12th standard. So, but cricket still helped me get into this college. So I actually got into college to play cricket. And when I started, when I went to the college with my cricket credentials, the two subjects I wanted to do were maths and physics. They were gone, so they gave me chemistry. So that was like the third choice. But actually, I think I was a pretty good chemist. So I was, good with balancing equations and the whole, different kinds of isomers, formal isomers, whatever. I mean, enantiomers, diastereomers. So I was pretty good at that. So I was really delighted. This is a beautiful campus also. And our head of the department, Dr. Gopalan, he had just returned from the US and he was writing a book. And I don't know what he saw in me. He said, Mohan, I need to transcribe. I don't have time to write. I'm going to dictate. Why don't you write? And so that was great for me because, and also, in 1984, he was actually paying me something like 20 rupees a week. So that's terrific. Okay. So he was paying me to write. And that actually really got me excited about because the way he would like narrate the story, it was not like doing hands-on research, but when you're writing a textbook, and in fact, I think I probably wrote parts of I didn't write it, actually, I was transcribing. This is like dictation and write down with a pen. I think one of the first editions of this book, I think I had a hand in that. So, but writing a textbook was so beautiful because you kind of present your ideas in a way that gets you excited about science. So at the end of it, my BSc, this man, P.F. Subramaniam, one of the most outstanding organic chemists I've come across in Madras Christian College, he said to me, Mohan, I think the future is in biology. So I went to this program in Baroda. This was the first time there was a national entrance test to do a course called biotechnology. And I got into the program and I met this man. And to me, he's the most influential person in my academic career. His name is Bharat Chattu. I don't know how many of you know him. He's actually one of the most amazing scientists. He had outstanding training in yeast genetics, plant biology, but because I came from chemistry background, when I went to to the department, he asked me to read a bunch of papers written by Tom Check, and this is about self-splicing RNAs. He said, I want you to learn, because he said, you didn't do any biology in your undergrad. I'm going to get you introduced to biology through chemistry. And then he gave me a bunch of papers and said, just come and talk to me about them. This was exceptional because I knew, nucleophilic substitutions and, SN2, SN1 reactions, all that. And then Chattu is telling me, all this is happening in biology. This is what happens in biology. This is what happens in biology. I was, really blown away by that. And so that became, then there was an obsession with biology. But we couldn't do chemical biology in his lab. He was a yeast geneticist and a plant biologist. So I actually worked a little bit with rice. I didn't work on yeast. But I was quite excited by some of the work Jerry Fink had done at that time. And so I had a plan, which was either to do plant biology or yeast genetics. This was going to be my future. And of course, opportunity has to align with aspiration. So you have to get a scholarship. And I got a scholarship in a place called Saskatchewan, Canada, which is a beautiful Midwestern part of Canada. And I came across this man, Sean Hemmingson, who had just discovered this machinery that helps proteins self-organize and self-fold, called the chaperonin family. So that's kind of, so then I thought, okay, let me work on this problem of how proteins fold. And then 2 months later, Sean Hemmingson, he came back from a visit to Oxford, and he told me, Mohan, there's this great story going around, cell cycle. Paul Nurse has isolated cell cycle mutants. Do you want to work on them? And Paul Nurse had been working on DNA replication and G to M transition, but nobody was working on cytokinesis. So I said, okay, I'll take a look at cytokinesis mutants. And that's the kind of mutants that cannot divide. And they would have multiple nuclei. So I worked on it in that lab. This was a plant biology lab. I started working on yeast, and I published a paper. I showed that tropomyosin was a component of the cell division apparatus, this actin binding coil protein. And I also identified two other proteins, profilin and a myosin light chain. At that point, I finished my PhD. And Sean could not continue to work on yeast because actually he was in a plant biotechnology institute. So he said, I don't know what to do. So I said, I'll find a postdoc where I can continue on this. So the first thing is, people always tell you to change topics for postdoc, but actually I didn't. I just went with the same. In fact, it was just an extension of my PhD. And in fact, all the 37 years or 38 years I've been in science have been an extension of my PhD. It's just that every 5 years, a new approach is used. Rather than go to a new system, I use a new approach. Okay, so then I went to Kathy Gould's lab in Vanderbilt, and there, this is Kathy, and this is Dan McCollum. And the important thing is, Dan McCollum and I, we were such great friends, and we used to talk about science every day. And so every morning, we would both get into the lab at 8:30 and look at the plates to see what we had. Okay, and then at about 10 o'clock, we would go for coffee or tea and talk about that day's results. And that was the most important thing. In fact, in science, you have to find somebody who you can discuss freely without any competition. And you should be able to do that so that you can continuously focus your ideas and make them better. And you have somebody who really will criticize you and not tell you nice things all the time. It's like, it doesn't help when people say, oh, it's great, it's great. Doesn't help. Okay, so this also, since I took the genes that I had cloned in my PhD, I also learned how to set up collaborations. I was a postdoc, and I got the two labs of Sean Hemmingson and Kathy Gould together to write a paper in Journal of Cell Biology. And that was really great. And in fact, all this was done without any great plan, okay? And we published a number of papers. And in fact, this paper would launch my entire career, okay? This is, after working with a little bit of Paul Nurse's mutants, I decided that the screen was not complete and many important components of the cell division apparatus were not represented in Paul's mutant collection. So I did a screen myself, and we isolated 16 new mutants, all of which were defective in cytokinesis. So I'm just going to summarize. What did I discover as a PhD student and a postdoctoral fellow? I think what I showed clearly was I established fission yeast as a model for actomyosin ring-dependent cytokinesis. And so the idea at that time was that this was heavily inspired by the sarcomeric situation. So actin filaments would be attached to the membranes with their plus ends, and then short bipolar myosin filaments would be pulling on them. And by this kind of a process happening throughout the cell perimeter, this you're looking face on, you'd have a centripetal ingression of the division apparatus. And what I showed was that fission yeast, here shown here, divides much like a mammalian cell using a contractile apparatus, and all the mutants I isolated were defective in assembly of the ring, and therefore they were defective in cytokinesis. Not only that, I told you that we had a number of mutants, and I cloned most of them when I was a PhD student or a postdoc, and then a few of them in my own lab, which I'll come to. And 3 out of the 12 that are shown here were cloned by other people. So essentially, we identified the entire toolkit required to assemble the actomyosin ring. And so I had all of this in hand. And there will come a point when you're a postdoc, when you have more ideas than you can do with your own hands. When that happens, you're ready to move on to run your own lab. Until that happens, stay in the lab where you are, keep publishing paper. So this question has driven my research for the last 30 years, which is what mechanisms generate force or tension, as a physicist would say, to divide a cell into two. So I had to find an independent position. And I just want to tell you that every scientific conversation you have is actually an interview. Okay, I'll tell you why. So I was actually in Cold Spring Harbor in October 1994 because my mentor, Kathy Gould, actually had gone on maternity leave, and she was supposed to teach the yeast genetics course. So she said, Mohan, why don't you go and teach that course? So I went there, and there's this scientist who I don't know how many of you recognize, Venkatesan Sundaresan, very, very famous plant biologist. So Sundar used to work in Cold Spring Harbor, and Sundar was a late night person. I was a late night person. So we just met one day around midnight, and he said, tell me what you're doing. And Sundar is such a modest guy. I said, Sundar, who do you work with? Because he looked so young at that time. He said, I work in my own lab. And then he said, tell me about your work. And I told him, all the cytokinesis gene identified. And then he said, 24 hours later, same late night, he said, Namhai Chua, who is in Rockefeller University, wants to talk to you because he's starting a new institute in Singapore and he wants to offer you a job. I'm like, what? I mean, then next day he called me. He said, Mohan, I've been following your papers, the 3 papers. And Nam's one of these people who used to send postcards asking for reprints. I don't know how many of you remember those days. So he said, I've seen your papers. And, there's this institute I'm starting. You're the kind of person I want. He hadn't, that's it, a phone call. And right. So it was also easy. And the beauty was that, so it was in Singapore, core-funded institute, PhD students were going to be given to you, common equipment. But everyone told me, Mohan, there's not much going on in Singapore. This was actually 1994, 95, before A-STAR, before National Research Foundation, before the universities were investing money, government was investing money. It's very, very early days. So at that point, in fact, I visited Singapore, and Sundar actually decided to go to Singapore, in fact, to the institute as director. And I met these 2 guys, Uttam Surana and Bill Chia, who also many of you know. In fact, a number of people here know both Uttam and Bill. And I just thought, these are the colleagues I want to have around me. It doesn't matter what other people tell me. Actually, I was in a Gordon conference. It used to be called Molecular Cell Biology. That conference doesn't exist anymore. And many people came up to me, many prominent names. I had actually published 5 papers from about 6 years as PhD student and postdoc. Mohan, why do you want to go there? Why don't you come over here? Why don't you come over? And somehow I just felt these colleagues are more important. And I just felt that if I can talk to somebody and if they can bounce off ideas and I can bounce off ideas, and I thought that is what I needed. So I decided to go to Singapore. I joined the Institute of Molecular Agrobiology in 1997. Within 3 years we had 8 or so papers published. But within 3 years, so it was just 3 years, and then the government decided through some politics that in 2000 the institute I joined was going to close down. It was actually been only 5 years since it was started, and they were going to close it down. So at that point, a company called Temasek Holdings, which is a government's investment holding arm, they bailed out this institute and said, okay, we're going to start a new institute called Temasek Life Sciences Laboratory. Except that most people did not trust going to a new funding agency, starting a new institute. And so, but actually I did. And the other people went to IMCB. So people were given an option to go to this Institute of Molecular Cell Biology. I decided to stick in this institute. So at the age of 35, I took over as director of this institute. And this was, and then actually after 2 years, we recruited Bill Chia as full-time director. So then I became deputy. So the question was, is it bold or is it stupid or is it naive? And actually I felt it was an opportunity because I was young. I could take the chance. If it failed, I could actually go back to being a PI somewhere. So that's what I did. And this was great because this gave me the opportunity to establish a scientific advisory board with people like Paul Nertz, Randy Sheckman, Elliot Meyerowitz, Rich Losick, Liz Roberts, and so on and so forth. I could recruit. So actually, I helped in the recruitment of 2 directors. One was Bill Chia from 2004 to 8. He was the director, and Steve Cohen from 8 to 10. And over this time, we recruited 25 PIs. we established a PhD program, and also we established the guidelines for running the institute. So at that point, actually, one important thing is that there comes a point you would be in power and you should want to give it up. And I felt that at the age of 37 was not the right time to just like go and do admin the rest of my life. So I decided that I would end up being a deputy. And I actually focused on my science. Being a deputy with Bill or Steve was just an amazing experience because these are, two of the brightest people. Drosophila people would know Steve and Bill very well. So, but actually, there's one job I wish I had gone for and I didn't, and I'll be happy to explain to people what that is later. So, what did we do in Singapore? So, we showed that. So, in Singapore, I told you I had all the mutants. In Singapore, we took that further. Lots of imaging. We could show that actin rings were assembled from cables that were drawn into the ring, as well as new de novo actin filaments that were made. We found that cell wall assembly was important for actomyosin ring constriction. So here's a cell wall mutant that does not constrict the ring. We actually developed an in vitro system. We could isolate the actomyosin ring in its entire complexity, add ATP, and make that ring constrict. That's upon ATP addition, the ring constricts. And finally, we also set up a synthetic biology platform where we could assemble bacterial cytoskeleton inside yeast cells. And so, but I would say more than the papers that we published, and I also got the medal from Singapore. What I cherish most, what I think is great, is I trained about 40 people at that time, 40 totally PhD students, postdocs, and technicians. 11 of those 40 are principal investigators, and many of these people. Naveed, Srini is in NICER, Mithilesh used to be in TFR Bombay, Anup is in Ashoka University, Sadanand is in ICER Trivandrum, and there are the Sneshka is in King's College London. Two of them are elected members of EMBO, four of them are Wellcome Trust investigators. So kind of everything was hunky-dory, going great. And, but I just felt that I need to get out of the comfort zone. There are pros and cons. The pros, of course, where I was, there was no grant writing. Okay, I didn't say that. There was no grant writing. I could get a group of 10 people, 6 postdocs, 2 PhD students, 2 technicians, all funded by my institute. It was just all coming very easy. But I felt my research was very repetitive. It's almost like, there was a formula and I got tired of it because, yeah, we were publishing in good journals, but it was very, very repetitive. The con is, so like I said, research. So I wanted to explore, new approaches to solving cell division. And so then I also was wondering, because it's a young institute, even at that time, I was like, oh, what would happen to my institute if I left? And this was kind of a crazy thought. And Mike Sheets, who had just come to Singapore, he said, Mohan, institutions survive, people don't. If you're unhappy, go. So I was like, Mike, that was great advice. And so I started looking for jobs. And so the question is, why UK and why Warwick? In fact, this time also cricket came to help me. At this time, it was not me, but my son wanted to play cricket. So he said, I'm not coming to America or Canada or anywhere in Europe. It's only UK, Australia, or New Zealand. What? Okay. So then we decided to go to UK. Again, I had universities giving better startup packages, but I went to Warwick again because of colleagues. So I want to repeat this. Startup packages are important, but not that important. Who is going to be around you, your colleagues? They are going to define you for 10, 15 years in what kind of research you do, what kinds of collaborations you set up, and so on and so forth. So look for colleagues. That's more important than startup package. 2 million startup package is not going to last it's not sustainable. It'll end, and only colleagues can sustain you. And so, I moved to UK and I ended up getting grants from Wellcome, ERC Advanced, so on and so forth. But I would really, credit Mike Sheets for that, because he had moved to Singapore to set up the Mechanobiology Institute. And the style of thinking that Mike had was so influential, and nobody in the cytokinesis field was thinking like that. I just took his ideas and wrote it in yeast, and I got all the grants. So essentially, I keep telling Mike, this was like just simply taking his cool ideas that he had been using in mammalian cells for force measurements, chemistry, just, transplanted them to yeast, and we got the grants. So the point is, you have to change approach once in a while, okay? That's the other thing. And what did we achieve scientifically in the UK? So in fact, we did a lot more of in vitro experiments. We could say how turnover was affecting actomyosin ring constriction. We could reconstitute actin filaments, curved actin filaments on supported lipid bilayers. We were able to purify yeast myosin and make it move actin filaments, yeast actin filaments, and we also solve the structures of certain actin mutations. And this here, I had this incredible team. Saravanan, who I think many of you know, he's now in Indian Institute of Science. Darius, who was with me for a short while, he's in Warwick now, and Clyde was still with me. And we also developed lots of methods. We developed this monomeric Stegall fluorescent protein, which is incredibly photostable. We developed methods to purify actin isoforms. And we also had a foray in understanding archaeal cell division, the Asgard family archaea. So this was all going well again. And then there was a major challenge, which is all my grants were running out and new grants were not getting funded. So in fact, the first 4 out of the 5 grants I put in between 2014 and 18 got funded. 5 out of 5 between 2020 and 22 did not get funded. Okay, so and you have to remember, administration can quickly be judgmental. It doesn't matter how successful you are, whatever. Suddenly they'll think you lost your interest, or you're just becoming repetitive, or you need to be thinking more about your research. Maybe you're thinking about other things. These kinds of things will come up. So I would say, everybody will give advice at these times. They're all well-meaning, but not right for you. Listen to everything, but trust your own ideas. Okay, and the recovery was, in fact, adaptation is the key. So my first 4 grants were all as individual PI. The last 4 grants have been team grants between 2 to 6 members. 4 out of 6 grants in the next period, 2023 to 25, have been successful. But I think my science itself was getting very collaborative. So, and the style of funding was also kind of going towards team science. But what I would say is make sure that the team grants that the team science grants that you're writing support your core interest. It's not good to just do something because money is available, okay? Because you won't do a good job. So what is next scientifically? In fact, we have started working on this fungus, which is called Aureobasidium pullulans. It has an incredible lifestyle. It can grow by budding, fission. It grows as a hyphae. It actually has a meristematic mode of growth. In addition, it's also exceptionally resilient to changing environments. And we also developed this model for cytokinesis, where we have a push-pull mechanism where myosin and cell wall synthesis are coordinately controlling cell division. And this is the kind of thing that we are testing now. Okay, I've also been in administration in Warwick, spent 8 years as pro-dean for research, 3 years as director of Institute of Advanced Study, and in the last year and a half or 2, I've been deputy pro-vice chancellor for research. The most important thing again is that I helped in recruiting the best people. One of them, this is Tim Saunders, this is Jitu Mayer, and also set up lots of international partnerships with A-Star, with WashU, St. Louis, and MCBS. I also helped set up an undergraduate course. I'll tell you about that if anybody's interested. Lessons, I'm going to summarize quickly. Only do the science you enjoyed and do it right and rigorously. Impact will come. Don't bother about the impact. In fact, I'm going to give you advice which is contrary to many other, don't look for impact. Do a really good job. If you do a really, really good job, the impact will come. Good, clean science will have, value, someday. And, people talk about GPS and Einstein and all that. I'm not going for 100 years, but, we can talk about that also. Surround yourself with the very best people who are better than yourself, and don't be insecure about it. I actually managed to have such outstanding people, both my colleagues and my lab. Actually, like I would say, I don't want to put a number, huge number of people in my lab who are far better than I am. Okay, and learn from them and support them. Science is about trust. So when you start your lab, you have to find multiple ways to double check results that young new trainees might bring results to you. You don't want to feel them, you make them feel bad, but you have to still make sure the result is correct because you're going to put your name on it and you're the corresponding author. Okay, find interesting, innovative ways of asking them to repeat experiments, not I don't believe it, do it again. Okay, but there are many other interesting ways you can say that. Time's important, truth's more important. Okay, so if there is a competition, don't worry about it. Yeah, sometimes you have to, rush, but actually getting it right is far more important. Okay, this is actually copied from a friend of mine. Write grant applications to do research. Don't do research to write grant applications. Okay, so you see the difference. Yeah, because a lot of times people just do research so that they can get a grant. Okay, perhaps the most important thing I would leave you with is that the people you train are your biggest contribution to science. It's not the papers. And legacy of n equals 1 doesn't work in science. In art, you could be a Michael Jackson or somebody and not train musicians. Doesn't matter there. In science, if you don't train people, it doesn't help. You need to train great people, and people should take things forward. Okay, be generous with the lab, and socials help immensely. The last is don't shun administration. If you do, then you lose the right to complain. Okay, you cannot complain about administrators if you don't participate in administration. Okay, and but don't let that don't let the researcher in you disappear by being an administrator, because sometimes that's what happens. You get an overdose of admin and power, and then the researcher in you sort of slowly disappears, and that's not good. You have to know, actually, the medium, the water pH is not good, because if you want transformants showing up in 3 days, then your media better be right. And if you don't know that, you can just go to the lab and say, where are the transformants, where are the transformants? So you have to know the lab's routine problems. And last but not least, if you end up in administration, think of all other PIs before yourself. You should be thinking about yourself if you are an administrator. Okay, I think I'm going to stop. I don't have a shlok, but I'm because I think, but but I think I can kind of, yeah. Okay. But thank you very much. Yeah. Okay. Yeah. All right. Yeah. Okay. Yeah. Can I get some water?
Thanks, Mohan. Uh, we'll take a few
Yeah.
questions. Yeah.
Thank you for the lightning talk. As a, PI yourself, and since you recruited many PIs, I wanted to ask, apart from the technical skill sets that you expect from them, what are the soft skill sets that you expect them? What will make the PI grow and the lab grow?
Yeah, I think the main thing is to see how they'll fit in the environment that you have. And they need to bring something new. They should be interested in people's work. And they should also be interesting to talk to. So it should be, you should be interesting to talk to, and you should be interested in other people's work. And and then, obviously, they have to take you out of, you don't want three people working on yeast cytokinesis. So you want somebody who will take it to a different direction, but still maintain synergy. Because if you want, let's say, we have a Center for Mechanochemical Cell Biology. If I want to work, like if I get somebody who works on, DNA damage response, then it has to be tied to mechanobiology somehow. It shouldn't be, the checkpoint signaling, that kind of, because then it gets too far outside. And there would be a different space for that. But what you have to do is to make sure that that nucleus and the approaches they take would be exciting to the person coming in, but then they would provide something else to that group. And also be a good listener, good collaborator, not be too demanding. Like you have to ask for what you need and all that, but not me. Yeah.
Thank you, sir.
Yeah.
Hello, Mohan here.
Yeah. Hi.
I'm Shikha,
Yeah,
and very nice talk. I have a scientific question. You have shown that when you were adding ATP, you were seeing a constriction, and then you have shown structures wherein you have solved the structures of actin mutants, you said. So, do you see a monomer, multimer transition as you add ATP? Like, have you checked that?
No. So the ring is a pre-assembled structure. So I didn't have time to go into that. We take a cell which has a ring and isolate it, and then add ATP. So it's a fully assembled ring which is poised to constrict. Yeah. And once you provide ATP, in fact, all the phosphorylation, everything has happened. So once you put the ATP in, then the myosin motors are in fact in a Reiger state. So they are actually attached to actin filaments, because without nucleotides, they're attached tightly. And then ATP comes in and then gets into myosin, and then you go through the cross-bridge cycle. So it's not about actin polymerization. The full actin polymerization has all happened.
Hello, Mohan. Thank you so much
Yeah.
for the talk. I had a question more on the mentorship style. So, given you have diverse experience working with people in different countries and geographical locations, over a period of time, like how mentorship and training the mentees has worked, or what are the different mentorship styles which would be something you can suggest to the early career scientists? Thank you.
This is a very interesting question. I think you should ask my lab about it, because I mean the first 4 or 5 years, I was the first to go and look at everybody's plates and results and pretend not to have seen it. Okay, because yeah, but then people started doing secret experiments, because so they would write different things on the plate so that I don't. They want to come and tell me the results, and they want to be the first person to tell me that excitement, not. So you have to learn this, but at the same time, we are all curious people. We want to know the results. So, but that joke aside, actually, the lab was. I don't know. I just felt that the lab environment, you know, running a lab for 30 years almost, I think that we have had only one authorship issue, like literally one in 30 years, and over 100 papers published. And so I think traditionally, I think the lab has been always been a place where people felt that they were appropriately rewarded, and people could uh, there was a lot of flexibility, and so I mean the thing is, right, you have to have zero tolerance for certain things. Okay, sexism, racism, all these things, you have to have zero tolerance, not even jokes. So that way everybody, you cannot have a group of people making jokes about another group. And so you have to make sure right from the beginning that the culture of the lab is such that everybody is integrated, and you make people feel like they're part of the group. And and that's all I can say. And I think um, it's just about how you how you treat others, and you can be critical, but at the same time you can be nice about it. And people make mistakes, and when people make mistakes, you just have them realize what it is, rather than just like be in their face. In lab meetings, try and be patient. I mean, there's lots of things, right? It's all about treating people the way you want to be treated. I mean, you would never want somebody to be shouting at you, or like telling you in front of the whole group that your experiments are bad or something. So it's just I don't have a great answer, but it's just that treat people well, and have zero tolerance for some things.
Mohan is going to be around with you, but we'll now start with the breakout
Yeah,
sessions.
yeah,
Thank you so much, Mohan.
yeah.